welcome everybody to Science at the Source. My name is Hilary Marusak. I'm a neuroscientist by training, and I study, we were just talking offline, THC and mostly in humans, which is something different than what we'll talk about today. But I'm very excited to talk with Rodrigo Klein, who is our special guest today, where we're going to talk about some of the amazing work being done at the University of California at Irvine, or UC Irvine's ICAL, or the impact of cannabinoids across the lifespan center, where we zoom in and we talk about how cannabis and cannabinoids can really shape brain development across the lifespan. So, Dr. Klein, thanks so much for joining us.
Thank you, Hilary. Nice to meet you.
Nice to meet you as well. Can you first start by just sharing a little bit about yourself and how you ended up studying THC and neurodevelopment?
Sure. I'm here in California at the UCI about 10 months. I finished my PhD. I did like all my studies, all my masters, my PhD back in Brazil. I'm from Brazil. And I finished my studies studying like the impacts of different substances. My main focus was the impact of acetaminophen on neurodevelopment. I finished these studies and I continue to have like this profound interest in the impact of substances on how environmental um, can contribute to alter neurodevelopment. So I observe in here, Dr. Piomelli has this phenomenal work with how cannabinoids can alter neurodevelopment as well with the ICAL project. And I contact him, and then we start to collaborate. And then I join his group for about 10 months now. I joined the lab. And then why I have this interest in cannabinoids specifically is because in my previous work, I have this observation that the acetaminophen also interacts with the endocannabinoid system. So I was studying this system for a while, and then disconnects in this manner. And of course, THC is a substance that is very popular, very common. Of course, in here in California is legal, but in many states in the U.S., in many countries around the world, it's illegal substance. So where is illegal, like in the places where is illegal, it's the most common illicit drug that is used by the population. So it is a concern if there is an impact on neurodevelopment. So that's why we are interested in studying this.
Yeah, that's fantastic. And as I think CC Hillard, another giant in the field, along with Dr. Piomelli, says, all roads lead back to the endocannabinoid system. So it's really interesting to hear that you are working on another drug class and it had involvement or impacts on this system. So that's fantastic. And I'm glad that you're finding it interesting so far. So I know you're relatively new to the cannabinoid field, but what have you learned so far about how THC affects the developing brain? And then, in your opinion, what are the big unanswered questions?
Yeah, so how it can affect the neurodevelopment it has the potential to alter the neurodevelopment in many ways. That's because like the THC, when we say THC, is the main compound, the main psychoactive compound present in the cannabis, right? So is the responsible to doing like the psychoactive effects of cannabis. So and it binds to a receptor, what we call like a cannabinoid receptor type 1. That's the main responsible for the effects of cannabis. And this receptor is expressed on even on very early stages of development, like on the brain of a fetus, and even early on development, like in the first days of after conception, is already present on the fetal tissues. So if this receptor is present even in these first days after conception and in the fetal brain in the perinatal perinatal period. It is involved in many different steps of neurodevelopment, like neurogenesis, like migration of neurons, like synaptic pruning, in different steps of neurodevelopment. Then we know that THC, by binding to this receptor, can alter these neurodevelopmental steps. So we know that it has this potential to alter neurodevelopment. What we don't know yet is what process it actually alters. Like it alters neurogenesis. Does it alters synaptic pruning? In what timing? Like, is it right after conception? Is it during adolescence? Is it during childhood? So what are the critical steps of neurodevelopment that THC affects the most? What we are studying in here in the ICAL is adolescence. And why we are studying adolescence? Why is this a critical period? We have like a couple of questions in here. Why adolescence? First, we have like the psychological vulnerability of adolescence. We can call vulnerability, because in adolescence we have like this risk taking and novelty seeking of adolescents, right? So adolescents want to try new things. They want also to assume some risks because they want also to try new things. So they want to explore more. They want to also engage in new activities, especially if like there are some kind of a social environment that want to push them to do this kind of stuff, right? So all of this makes it easier for them to even to try drugs. So cannabis is one of them, and even in some places that cannabis is a drug that is of easy access. So it's a drug that is easily accessed by some populations. Then cannabis is a drug that they can try. And adolescence is a period that the brain is highly plastic. And when we say plastic, is actually when the brain is still doing this neurodevelopmental process that I mentioned. So the brain is still developing, the brain is still being changed. So the brain is still doing synaptic pruning, the brain has fluctuations of neurotransmission, all of these steps that THC can bind to CB1 receptor, and this activation, this repeated activation of CB1 receptor can alter this neurodevelopmental process, right? So that's what we call a window of vulnerability, a window where things can go wrong. And that's why we are interested in studying this.
Yeah, that was beautiful. I took down some notes because I think you just very clearly laid out why adolescence in particular is so important for studying THC effects, not only from kind of like adolescents being hardwired, it's literally in their neurobiology to risk take, and the
Yeah,
social context is so important. And then also pointing out a window of vulnerability for not only brain development, but psychiatric risk. And then
exactly.
very nicely pointed out that there is potential, like a very clear molecular mechanism by which these things could alter very fundamental brain developmental processes. So thank you so much. You just connected a lot of different kind of perspectives. So talking about really the cellular, molecular down to the receptor level and how THC, that active component of cannabis, could interact with that system all the way up to behaviors. So how do you kind of connect those dots in your own studies? And you mentioned you work with animal models. So what does that look like for you?
Yeah. So we have many unanswered questions in this, like connecting all the dots until we have like CB1, like THC binding to CB1 receptor, until we have like an altered response after this. So we know, for instance, like in association studies and in humans, for instance, we know that THC during adolescence can lead, for instance, to altered cognitive development. We know that there are some psychiatric risk after exposure to THC during adolescence. But like doing this bridge, like THC binding to CB1 receptor during adolescence and then linking to a later behavioral, psychological, psychiatric alteration later in life, it's really hard, even in animal studies, like to do like this molecular alteration in here and then a behavioral association. What we know from our studies in here at the lab, and we are trying to do so because it's really important to know the mechanisms of how THC alter these fundamental processes until it leads to a behavioral change, is that we are observing that a critical step for these behavioral changes to happen is CB1 receptor activation in microglia, actually. So that is kind of a surprise, because if you imagine that CB1 receptors, usually we're talking about neurons and so on. So neurons are a huge population in the brain, right? But they are not the only cells in the brain. In the brain, we have multiple types of cells, right? So we have neurons, we also have glial cells. In the glial cells, we have astrocytes, and we have microglia. Microglia, we are talking about these cells that are the first line of defense of the brain. They are like our leukocytes in the brain, the white blood cells in the brain. Some people say like they are the macrophages of the brain. So in our studies in here, we are observing that this microglia, by CB1 being activated in this microglia repeatedly, this microglia enters in a state where they are less responsive to stimuli, like for instance, immune stimuli, even psychological stress stimuli. So this microglia become hypo-responsive to stimuli, and they are very important to respond to environmental stimuli in general. So we think like right now, our hypothesis, and we are testing this right now, is that this microglia can be actually like the first molecular and cellular response to THC that, being constantly activated by CB1, will enter in this hyper-responsive state, and this will alter how the cell interact with neurons. So it will alter how neurons are being activated or not activated, how microglia can do this synaptic pruning that I mentioned, that is very important during adolescence. And then the microglia will not be able to do the correct synaptic pruning in brain regions that are very being still matured during adolescence, for instance, the prefrontal cortex. And then without this neurodevelopmental process that are dependent on microglia, then this leads to behavior alterations later in life. That's our hypothesis right now. These are still being tested, but it seems the case right now from the data that we have. Other groups have observed some in the same path. Of course, this is not the only case. We are observing just one case, because THC, again, the CB1 receptor are all over the body, not just the brain. They are in your liver, they are in your blood cells, they are all over the body. So we are just trying to see one mechanism. There could be many other that could lead to behavior alterations or even immune alterations or even more that we don't even know yet.
Yeah, and your explanation did such a good job of just highlighting how much we don't know. And I also love that you led with, in humans, we see that adolescent THC use is associated with psychiatric and cognitive effects. So you're bringing that back to the lab to see how does that actually work at the level of cellular molecular processes. So I'm super excited to see what you guys find. Maybe next time I interview you, you'll have some more data to talk about. But yeah, I want to hear a little bit more about like what are your plans for the next couple of years? Like, what are you most excited about at UCI and beyond in this line of work?
Yeah, so what I am excited in here is to finish these experiments and to see what is happening, because of course I'm talking in here, and I want to conclude more. Then of course science is always like this, we always want to conclude, but it's never the case.
More questions always, right? Than
Exactly,
answers.
probably I'll have way more questions than I I have now.
Right.
But I do want to finish these experiments to be able to share with you at least the results, even if I have more questions. Uh, I want to collaborate in here because we have such a great team in here around, like in the campus. We have such a group of experts in microglia, in molecular biology. We are working together to trying to solve all of these paths that is leading to these alterations following adolescent THC exposure. And also trying to find different effects of THC on the whole organism. Right now, we are focusing, of course, on the brain, but we are also having this click that THC is affecting all the organism, like is affecting the blood, the the metabolism, the sleep. So we are maybe having this broad view of how is the organism responding to THC. And that's something that I wanted to explore. Another thing is that I also have this itch in my mind that different kinds of and maybe doing this translation, different kinds of people would respond different to THC. And let me explain what I just said. If we are investigating the typical neurodevelopment, the effects of THC on typical neurodevelopment, but what would happen if we have an atypical neurodevelopment? What would be the THC effects on the atypical neurodevelopment? So we are observing that in typical neurodevelopment, THC is mostly doing bad things, to summarize, to be very straight. But on atypical neurodevelopment, would that be the case? So that's a question that I personally have, like that I'm interested in investigating, because we know that those pathways of neurodevelopment, they are very distinct. So I'm not so sure if it will be the case of doing just bad things.
Yeah, and this is kind of a theme too with all of my interviews with the amazing people at iCal is, if you say is THC good or bad, it's like, okay, for what, for who, for what age, for what demographic. So that's why I love what you're doing because you're really pointing out that those things matter, that the age of the THC exposure, the organism itself, the type of organism, all plays a role here. So Amazing. I appreciate you sharing that. I want to end with some advice. So you are a postdoc. You mentioned you recently, completed your PhD, moved from a different country to the United States. So what, young advice would you have for young scientists who are considering making moves like that or considering studying THC or cannabis effects?
Um, I don't feel in the position to doing any advice to anyone, but I don't know, maybe studying hard, because that's what we do. We have to. Like right now, we see a bunch of people just in my view saying things that they don't know. Like in the moment, I sometimes feel bad to see people in positions of power um, saying things that we as scientists do not agree at all. And that's something hard. So we just need to study and keep doing our work. And that my advice is just to keep going and to study hard and also do what is important to you, like to in your personal view, keep going. Do not like see how others are doing, like do in your way. That's really important to keep moving, like and to persist in that, because otherwise maybe you give up. Like that's my personal.
I thought that was great. None of us have all the right answers, but I thought, you have such an amazing journey and you're on the right track. And I didn't know if you were going to comment on, some of the recent situations with people misinterpreting some of the science that you yourself have done. But I think you're exactly right. Just keep doing the good work and being motivated by the things that motivate you. So
Yeah,
thank you so much for sharing. I appreciate it.
you're welcome.
And thanks for being interviewed today. It was a ton of fun.
Yeah, it was really nice.
All right.
Thank you, Hilary.
Thank you so much. I appreciate you joining.